Pancreatic cancer is one of the most lethal forms of cancer, primarily because it is usually diagnosed very late. Current markers are too insensitive and unspecific for early detection screenings. A research team has now introduced a new method that could lead to a significantly more precise and reliable diagnosis. It is based on the selective detection of specific antibodies in blood samples. Glycoptide probes detect tumor-associated antibodies in blood samples. In the journal Angiochemy, a research team has now introduced a new method that could lead to a significantly more precise and reliable diagnosis. It is based on the selective detection of specific antibodies in blood sample. Tumors produce certain proteins, tumor-associated antigens, that draw the attention of our constantly patrolling immune system and trigger an immune response. As a consequence, antibodies directed against the tumor-associated autoantibodies are formed, circulating in the blood at very early stages of the disease, which makes them useful for early detection. An international team has now developed an approach to diagnostic testing for pancreatic cancer that is based on the detection of such special tumor-associated autoantibodies. They chose to use autoantibodies directed against the tumor-associated form of mucin 1 TARMAC1. Mucin 1 is a heavily glycosylated protein, a protein with sugar components that occurs, for example, in glandular tissue. In many types of tumors, including pancreatic cancer, it is found in significantly elevated concentrations. In addition, the pattern of glycosylation is different from the normal form. The team's goal was to detect autoantibodies that are directed specifically against TARMAC1 and are a clear indicator of pancreatic cancer. Based on structural analysis and computer simulations of known antibodies against TARMAC1 SM3 and 5E5, the team designed a collection of synthetic glycoptides that mimic different segments epitopes of TARMAC1. They also made unnatural modifications to increase the chances of identifying autoantibody subgroups indicative of the disease. Researchers immobilized these model antigens on gold nanoparticles, achieving probes suitable for a serological assay. The diagnostic assay was validated with real samples from patients with pancreatic cancer and a healthy control group. Some of the nanoparticle probes could differentiate very well between samples from diseased and healthy individuals, demonstrating they detected tumor-associated autoantibodies. Notably, these specific autoantibodies displayed significantly better correct positive or false positive ratios than current clinical biomarkers for pancreatic cancer. Probes with smaller glycoptide antigens that correspond to only a single epitope gave better results than larger probes that mimic multiple epitopes, an advantage for easier synthetic production. A short glycoptide with an unnatural modification to its sugar component was found to be particularly effective for the detection of discriminating autoantibodies. This new structure-based approach could help in the selection of autoantibody subgroups with higher tumor specificity. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.